The need of large biobank studies like UK Biobank, Estonian Biobank, or FinnGen, they come from the need that we wouldn't treat patients as mean values, that we could move from the mean value towards an individualized treatment and prevention, meaning more personalized medicine or precision medicine, whichever nomenclature you like to use. And thinking of what biobanks are doing, they are collecting typically samples from a broad set of volunteers, which means that we have a broad set of diseases that we can cover. And by doing it in a somewhat disease agnostic way, meaning that we don't focus on just one or two diseases, we can start to understand the essence of diseases better, first from their genetic background by looking at the DNA. Also, the biobanks collect serum and plasma samples, so we can get an understanding of the metabolic and proteomic profile of the circulation. These help to understand the biology of the disease better. And by understanding better the biology, we can build new avenues for both prevention and treatment. Without new biological insight, there's no new treatment or prevention either. Well, that's really in the heart of what biobank research is about. As already discussed in the previous question, genetic information is one stepping stone to create the personalized background or personalized information of each participant. But if our genetic discoveries don't produce anything else than a list of genes, we could maybe say that we have failed. So the question is, how do you move from a genetic variant that you discover in your association analysis? How do you move towards understanding the biological consequences of each variant? Proteomics is one of those avenues, and especially what we hope to find from proteomics is new biomarkers. And having biomarkers that either catch a disease process very early on or are good in monitoring disease progression, which has been traditionally the case, for instance, for cancer markers, that they are used as treatment to monitor the progress of treatment or progress of disease. But having a larger set of proteomes to choose from, that which of those proteins might be the ones that could be further developed to markers that can be used for either preventive purposes, so that we understand that how to tackle early things, something which would be much better than PSA, which we use currently for prostate cancer screening, but are not very happy with it. So proteomics provides a tool towards those steps. Right, so if we think of again, from moving from initial, say, genetic discoveries, which is the field I'm working in, towards understanding how that information can be translated towards treatment. Proteomics is an important potential tool there, and that's why there is such an excitement in the field that to measure proteomics in sufficiently large sample sets, that we could understand that what is the consequence of a genetic variant, which are the proteins, an alteration or a change in a protein or original genetics code, which are the changes we can see in our proteome. And possibly we think of drug development, possibly to understand from that data that what are potential adverse effect risks if that particular genetic target would be addressed. And again, as a very promising screening tool, we hope that proteomics is one of the key instruments that could guide us towards an understanding of these pathways that result either in potential adverse effects or efficiency of a drug. So if we think of medical diagnostics for decades, even for centuries, so it's based on understanding biochemical changes in the human body. Classically, we do this in the hospital laboratory using clinical assays like lipid assays or some protein assays to understand if they are signs of a given disease or disease's state. And thinking of proteomics, so this is again when we start from a screening approach and understand which proteins change in a disease state, we can develop those findings into functional biomarkers, in markers that we can really use in a clinical setting. So thinking that proteomics provides a tool to identify what can be brought into a clinical setting with all the clinical demands that a laboratory or a clinical laboratory measure typically has. We come back again to the biomarker thinking that obviously we think of the entire healthcare system, the most efficient way would be to get prevention more efficient than it's now. That would be probably the cheapest way, most likely the cheapest way, and also most efficient way. Because as we know, once a disease is already diagnosed, in many ways, we have already entered into a much more difficult concept. And thinking again, what could be markers, again, those biomarkers that would help us to understand risk profiles. Whether that is in cardiovascular diseases, something where this tradition is really long. We have measured lipid values for decades and decades, and those provide us early tools in the disease process to understand that when preventive treatments or preventive other measures are taken. Proteomics will very likely have the power to identify again new clinically applicable biomarkers that could be used to understand early whether that's cancer risk or some other risk. Time will show that proteomics, with the ability to measure a very large number of circulating protein components, provide a very exciting screening tool, again, to identify the ones that we would then use in a clinical monitoring setting for prevention or disease progression.